that make up the whole individual. Say sa isa ka cell, whole genome na or say nuclear, that makes up the whole individual. Oh? Now, kung imuhan ng kuhaon so by join together all the DNAs, it could measure around 2 meters. That is for the whole genome. And remember, how could it be accommodated in a very small nucleus in a microscopic cell? 2 meters. How could it be? Now, imagine a thread. The thread may be compared to as the DNA. Now, how can you place the thread in a 2-meter thread in your small packet? Lukuton ninyo, diba? You roll it. So, i-roll ninyo kanang plastic or it could be maybe a stick. So, place it after in the small packet. Now, that is also true for our DNA. It's very long. In fact, if there is around 2 meters for all the DNA within the genome, now sa isa ka cell, okay, that is only for one cell. And how many cells you have in your body? Billions. So you times billion, so you multiply it into a billion times. So it is very, very long. Accordingly, you can reach up to the sun several times, back and forth, Earth, Sun, Earth, Sun. Kung imo na i-join tanan together, ang tanan DNA sa whole body. So that is how marvelously you are made. So you are marvelously created. There is a designer for that. So you are not here by chance. Okay? With that structure only, with the structure of DNA, then we could say that there is a designer. Okay? Now, so you have now the histones. Now, that, say, the protein where this DNA is rolled is called the histone. So you have the histones 1, 2A, 2B, and 3 and 4. Okay, you move forward. Para di ka katulungan mo. Okay. So, 1940s, it became clear that the DNA is the genetic material. So it's not the protein. Because what comprises the chromosome is, of course, as what I had said, DNAs and proteins. Now there were, speculations before that it was not the DNA, which is the genetic material, and the protein. But as time goes by, with all those experiments conducted, it was proven that, especially in 1940s, that it's the DNA that is the genetic material. Now, you say genetic material. Now, a portion of this DNA that codes for a protein is called gene. And gene is the hereditary unit. Okay? Are you with me? Okay. So, you have here. Now, this DNA can be switched on and can be switched off. Now, this explains why there are some features or say phenotypes or appearances which can only say be observed in a particular stage of, or say in a particular age of a person. And say for example, usually Alzheimer, or say usually this gene for Alzheimer would be switched on in the late stage of a person. Okay? Or say for example, another example is the male secondary characteristics. What causes male secondary characteristics? Why it can be observed during puberty stage? Because it is caused by the production of a hormone called testosterone. And testosterone is actually a hormone, of course, which is the result of the activation of the gene for this hormone, testosterone. Okay? Now, this gene is said to be switched on at puberty stage. That's why there is no secondary male characteristics. What are those secondary male characteristics? Muscles, mga hairs. Oh, you cannot see that in the childhood yet, no? But you can observe that in the puberty stage, because at that time genes are said to be, for that is said to be switched on. Okay, so though only those genes necessary for a particular stage of a person is said to be switched on, the rest will be switched off. So in other words, when the egg is fertilized by the sperm to make up a zygote, that zygote is a single cell which is said to be composed of 2 sets of chromosomes. Now, all the genetic informations to become the whole you or the whole adult is already there in the zygote. But only genes necessary for the development of the zygote into embryo, then embryo into fetus would be switched on. Are you with me? And of course, after delivery, another genes would be switched on. So, because the switching on of a particular gene may trigger the switching on of another genes. But of course, if they are no longer needed, then they would be switched off. Now remember, we are regulated. There are chaperone molecules that regulate the different processes in our body. If you have supervisors, if you have chaperones in your dormitories, you have deans to supervise, then our body has chaperones also. There are so many supervisors in our body that regulate all the processes. And that is how marvelously you are made. Okay.
Ma'am.
Yes.
Ordain ako sa mga sulat sa board, ma'am. Gawanin niya po na pamisig sa kusina siya. Sa news trend sa last nga fertilization.
Ganyan na tayo. Wala pa na. Isa ta. Yan. Wala pa ta nag-take out sa fertilization. Okay? So, we have now the structure of the DNA. So, this is the hydrogen bond. This is the nitrogenous base. Cytosine paired with guanine, which is held together by a weak bond called the hydrogen bond. Adenine paired with thymine. Okay, so this is actually the DNA. So DNA can be, or say chromosomes, which comprises the DNA. So what comprises the chromosomes is said to be made up of DNAs, and this one is the protein. Mga histones na siya. Those are histone proteins. So mura sa mga yuyo. So they are highly organized. And this structure, this chromosome structure can form different, of course, they can have different forms. Okay, so you have there. So you can have these loops. Now chromosomes can only be, say you have here, different, another structure of the chromosome, the mini band, which comprises 18 loops, which are said to be necessary for the preparation to become chromosome. Now, chromosome can be observed only when the cell divides. When the cell is not dividing, what you could see inside the nucleus is the chromatin material or chromatin materials. Okay? So, chromosome, so that chromatin materials will undergo condensation sa mag-condense siya, mo-form din siya mga different loops, higher structure of DNA, hangtod they form stacked mean bands, no, to become chromosome. Okay? So you have here the differences of DNA and RNA. So there are three main differences. I mentioned that already. Okay. Now, how is this genetic information be transferred? Now, you have here the central dogma of molecular biology, of which so we have, say if this is our DNA, the DNA will replicate. So this is replication. So this is what we will be tackling, that one. And then out of this DNA, this will undergo transcription.
Translation.
So the product of transcription is the production of different RNAs.